This is a bit on the evaluation, question 5, and we've got an experiment here where we've got some potato containing the catalase and hydrogen peroxide, and then what will happen is hydrogen peroxide, so hydrogen peroxide there will get converted into oxygen plus water, and because oxygen is a gas, then we'll get bubbles coming up here, and then that'll give you more gas in here, and then you'll get bubbles coming out of there. And so you'll be able to measure how much air you've got in there, or actually the oxygen. And that's catalysed by our enzyme catalase in the potato. So things to think about in this experiment. I mean, overall, is it a good experiment? It's not bad. The experiment will work absolutely fine. And you've got a conclusion here. It says learner concludes that up to 41. So if you drew a graph of this, so this was temperature, and this was volume of O2, then 10 so 20, 40, 60, and the volume goes up to sort of 6, so 3, 6, 4, 5, 1, 2, then 2.3, that's down, it's down there. 20 is 4, so 1, 2, 3, 4. 41 is 5.45. 51 is 4.5. Going down. 59 is not point. Sorry, this is right down. So that's the shape of graph that they've got. And so the learner has concluded that up to 41, the rise in temperature increases the rate of catalase activity. That sounds good. And above 41, the enzyme is denatured and its enzyme decreases. So the conclusion made, it's okay. You can't argue with that. You can just, the criticisms we're going to make is how you make that sort of even better. So the data does support the conclusion. So data supports conclusion. But is the optimum at 41, or is it 42 or 39, or we don't know. There's only 41, the lower is 20, and then there's another 10 higher. So we could do with more readings around 40, 41 to find out exactly where the optimum is. So need more independent variable intervals around 41 degrees to give us the exact peak there. For example, every 2 degrees C or something like that. And we just need more points in general, so where does it decrease and where does it level off? We could do with more intervals generally, really. And then the various problems with the experiment, you can come up with all sorts. So these chips here, so potato chips, well, our question is, it doesn't say what size they are, shape, are they cylinders, are they slices, same potato. So those are a bit of a mystery. Or different potatoes might have different amounts of catalase in. Problems that we got with the the measurements. So readings are not repeated. So you generally do things 3 times and work out a mean. So we've got here, we've got no repeats, which is a problem. We've done halves there and different levels of precision there. So not all to the same precision, even the independent variable and the dependent variable. And here we're not exactly stating what volumes we've used, so you'd always look for volumes and temperatures and things like that. So what haven't they mentioned? So boiling tube containing hydrogen peroxide. What volume have we got? Because obviously if you have, that's our substrate. So more substrate will give us, in the previous question that you've just done, you're increasing the substrate. So more substrate give more oxygen. So that would be a problem. It says after 10 minutes the potato chip was added, and then it just gives the results that you get. So presumably they left it until the whole thing had finished, but we don't know. So how long? Then it repeats the step. So how long for the experiment? Question mark. When were the readings taken? There might have been some oxygen that escaped before this bung was put on. So they added the 2 together, then put the bung on. So there might have been some initial oxygen escaping there. So some O2 escapes at the start before the bung is on. Could possibly. So that might lead to inaccuracies. There's a point about the range. So this isn't a uniform range. So not a uniform independent variable range. So various little problems. You haven't got a lot of time. It's only 8 marks. So I would suggest you start off looking at their conclusion and see whether you agree with that and whether the data supports it. Then try and pick out lots of bits of the experiment. They'll deliberately left out things like volumes or temperatures or various little problems there.